Hi guys, wish I was actually talking to you in person, but we're going to do a redneck review today because I don't have access to the building. I don't have a dry erase board, but I have plenty of cardboard in the greenhouse, so you wouldn't want to see this setup. I have my cell phone taped to a hanging basket, but we're going to make it work because we have a test on Tuesday. The test will probably be open for an hour and a half. I'll put all of your information on Blackboard about when the test is and how long it'll be open and the duration. So you'll probably just be open for an hour and a half, hour 45 maybe, and probably for about a 12-hour period. So let's first start with our major biochemicals that we talked about. Remember, one was carbohydrates. So, you know, what you want to know on a carbohydrate, remember the basic form of a carbohydrate is CHO, carbon, hydrogen, oxygen. Remember we discussed a monosaccharide, which is a single carbohydrate, a disaccharide, which is two, and then a poly, which is two or more. We also talked about the importance on the carbohydrates between where the hydroxy is located at the number one position. Remember the difference between cellulose and starch. So, and then we talked a little bit about cellulose and starch, how cellulose is not able to be broken down except by ruminants, and how starch is a good source of energy. So go back and review that, make sure you know those positions and the difference between cellulose and starch. Our next one is our lipids. Remember, our lipids are our fats and our oils. We discussed the fact how saturated, unsaturated, triglycerides, so remember, or trans fats, sorry. So our unsaturated, those are liquid at room temperature. Our saturated fats are solid. Our trans fats, remember I said that those are fats that aren't even real. They're made somewhere in a factory, so that's your margarine. Those are the ones that have a tendency to clog up your arteries. But then we also discussed how lipids are important when it comes to chill injury on your plants. So if a plant itself in its leaf tissue is made up more of saturated fats or unsaturated fats, that has to do, and of course if one of them is liquid at room temperature, so you can see how when we have a cool spell, that it could actually freeze those tissues and then cause those tissues to either burst or dead, decaying tissue in it. And I think we looked at the example of the peach blossom and the strawberry bloom. So that's kind of what I want you to know about your lipids. Also remember that our lipids make up our phospholipid bilayer. So remember the bilayer surrounds all of our cells, all of our organelles, and they're made up of these fats. Remember we had two layers, so we have a hydrophilic and a hydrophobic. So the hydrophilic pulls water in, the hydrophobic pushes it, so that's what allows liquids to move in and out of the membranes. So if we talk about proteins real quick, remember proteins are embedded within that membrane and are adapted to bring special chemicals in or out. So for instance, an aquaporin is a protein that's embedded in our phospholipid bilayer, and it allows water to transport in and out of the cell. So on proteins, so our basic form of a protein is an amino acid. And remember, if we get larger than an amino acid, then we have a peptide. And then more than a peptide is a polypeptide. And we discussed how that simple chain of amino acid looks like a string of pearls. And remember we said that our secondary structures, or when you take that simple string and you either twist it around and it makes a helix pattern, or it's pleated and folded like a paper fan. And that's our secondary structures in our proteins. And then our proteins can also have tertiary and quaternary structures. So a tertiary structure is when maybe we have a circular helix and a pleated fan, and they're actually together, so there's more than one. And then the quaternary is when we have a multitude of these structures actually held together to make up some type of a molecule that has our proteins in it. So, and then nucleic acid is our other biochemical. Remember, it is our basic makeup of our DNA and our RNA, so that's one of the things that, it's what makes our nucleic acid important. I said that I had a list on there. Don't worry about the list of those. That's too in-depth for this class. You won't have to repeat those for the test. So I think that's all for our molecular, our biochemicals. So go back, review, remember that was that first section that we did. So then our next section is our section on photosynthesis. I hated that it's been so long since we talked about this because photosynthesis is sort of detailed. So this first one, we're going to talk about the light reaction of photosynthesis. Do you remember that photosynthesis occurs in the chloroplast? And within that chloroplast, we have these organelles that are called thylakoids. A single stack, like a single pancake, if you think of it shaped like that, is a thylakoid. An entire stack is a granum. And then this area out here, this liquid, is the stroma. So, and a single one is a thylakoid. So if we actually look, oh no, my tape fell. Sorry about that. All right, I got you back up. So if we look into the interior of the thylakoid, where photosynthesis actually occurs, it's happening within those membranes. So if we draw this, we know that this right here is our thylakoid. This area outside of that line, that's our stroma. So the light reaction of photosynthesis occurs within the thylakoid. So if we remember back to our equation of photosynthesis, we know that one of our input for the light reaction is water, right? So our input is H2O. So we take a molecule of water is taken into the plant. And it is, remember we talked about reduction and oxidation. Reduction is a gain of a hydrogen. Oxidation is a loss of a hydrogen. So our water is oxidized and we end up splitting the hydrogens, which we end up having some electrons from the actual oxygen itself. And that is done with that input of energy from the sun. So our first input of energy from the sun we measure as P680. You might go back and review the slides that talked about wavelengths of light and how we kind of measure those, which end. So our 5 to 600s are on the red, orange-red end. 300 is on the blue end. But for the light reaction of photosynthesis, we're on that red end of that spectrum. So the first thing is our water is oxidized. So because of the sun, and we measure that as our P680. So our second input over here is sunlight. Our electrons are energized by this sun. They move down an electron transport chain that is in the interior of the thylakoid, and as they move, a gradient is developed. This gradient pushes hydrogens through a pump. Remember, we have a pump there, and what we produce is ATP. So ATP synthase happens across the membranes of the thylakoid. So we end up right here producing ATP, which is one of our outputs of the light reaction. So then we have another input of solar energy that is absorbed. We measure that as P700. The P700, once again, excites those electrons, and we end up with NADPH. So the NADP gains through reduction, it gains a hydrogen, gains electrons, and we have a usable form of energy in NADPH that we're going to use later on in the dark reaction. So our second output of the light reaction is NADPH. Okay. Originally on our test, I was going to give you all sort of a blank version of this, and you're going to have to fill it in. Our test will be all multiple choice. So what I would look at, really concentrate on this little chart here. What are your inputs in the light reaction, and what are your outputs? Also, don't forget that the light reaction, we said, happens in two systems. So the first one, remember, they're labeled backwards. So this is photosystem 2, and this is photosystem 1. The photosystem refers to that input of energy in the form of the sun. And so we break those up. Photosystem 2 happens first. Photosystem 1 is that second input of light energy, is P700, and it happens second, even though that's out of order. So. All right, so the second part of photosynthesis we know is called the dark reaction, or the Calvin cycle. So where the first part of photosynthesis, the light reaction, occurs inside the interior of the thylakoid, our Calvin cycle happens to the exterior. It actually occurs in the stroma. So remember that the Calvin cycle is circular in nature. So we know that it turns in a circle, it revolves. So we have 3 steps to the Calvin cycle. So our first step is fixation. So if we remember back to our equation of photosynthesis, remember on the input side of our equation is water and carbon dioxide. So first thing we have is the fixation of CO2. So CO2, remember I said, is grabbed, and it's grabbed with the help of Rubisco. So that Rubisco acts like a puzzle piece and grabs a hold of our CO2 and actually pulls it into the chloroplast itself and into that stroma, where then it can metabolize it for the reaction of the Calvin cycle. So our input in the Calvin cycle, our first input is going to be CO2. So our second phase, so remember this is grabbed by Rubisco. So our second stage is reduction. So remember, reduction is we have a gain of a hydrogen and a gain of, of an electron. So in the light reaction of photosynthesis, we had those usable forms of energy in NADPH and ATP. So we use right here our NADPH is used in the process, the reduction of our CO2. So we have reduction of our CO2. Our output is right here, is we have a byproduct of oxygen, and we produce a molecule of glucose. So then our third phase is called regeneration. So our regeneration phase, its only purpose is to produce that Rubisco again, because once we've used Rubisco, if we don't have it, we're not grabbing onto our CO2. So the third part of the Calvin cycle is the regeneration of Rubisco. Which is a necessary step. So now we said how many times does the Calvin cycle actually have to revolve? to make 1 molecule of glucose? We said 3 times. So we are revolving 3 times to get 1 molecule of glucose. So our output in the Calvin cycle, or the dark side of photosynthesis, because remember we're still talking about photosynthesis, our output is glucose, and we have an output, a byproduct of oxygen that's actually produced. So I also want you to go back at the end of photosynthesis, and we discussed C3, C4, and CAM plants. So we said that I would know some examples of our C3 plants. So remember, there are plants that grow in cooler weather. So that's our wheat, soybeans, and the C3 plants. The majority of photosynthesis that happens every day in plants is actually C3 photosynthesis that is occurring. So remember, the C3, we have what is called photorespiration. So sometimes when we get hot days, the stomata, the pore on the bottom side of the leaf, will close. When that stomata closes, it traps this byproduct of oxygen in the leaf. What this oxygen wants to do is it wants to go back up here, and it wants to claim that active site on rubisco instead of carbon dioxide claiming it. So it interferes with the process of photosynthesis. We call that photorespiration. So that occurs in C3 plants. So C4 plants have figured out a way to bypass that. So examples of our C4 plants, these are our plants that like the heat, love to grow when it's hot. So that's our corn, sugar cane, all of our agronomic crops that like to grow in the actual heat of the summer. So our C4 plant, remember we go back and review those slides about what the internal makeup of a dicot leaf and a monocot leaf looks like. So the C4 plants have an adaptation where they have separated the oxygen and the CO2 with space. So it's how those mesophyll cells are around our vascular bundles, and they separate that oxygen. So when photosynthesis, when the Calvin cycle is going on, we have this byproduct of oxygen. It's pulled away from the cells where the Calvin cycle is actually occurring, so that that oxygen can't get back in there and compete with that CO2 for that active site on rubisco. Now, we also talked about CAM plants. So we know our CAM plants, they have a completely different adaptation. They have adapted to growing in extremely hot weather. That's our cactuses, our succulents, our euphorbia. And what happens, hold on. So our succulents have this very thick leaf. So what they have done is they hold a lot of moisture inside to the internal portion of this leaf. And they actually photosynthesize at night. So this whole process goes on in the evening so that they aren't opening those stomata and releasing some of that water through evaporation that they end up needing later on. So that for CAM plants to survive, they have adapted with a difference in time. So our C4 plants is a difference in location, and our CAM plants have adapted with a difference in time. All right, so the next thing we talked about in class was cellular respiration. So cellular respiration, what happens when we produce that glucose, the plant has that sugar, it's not quite enough energy for all of the metabolic functions that have to occur within the plant itself. So we have to go through cellular respiration to produce that ATP. So we said cellular respiration happens in the mitochondria because that's the powerhouse of the cell. That's where we're producing our ATP. So within the mitochondria, we said the interior space in the mitochondria is called the matrix. Remember, our mitochondria is one of our organelles that has a double membrane. So that internal membrane is called the cristae in the mitochondria. So our phases of cellular respiration are glycolysis. Glycolysis is the only phase of cellular respiration that does not occur in the mitochondria. Remember, glycolysis occurs in the cytoplasm. So it occurs in the cytoplasm. Glycolysis takes that glucose and breaks that glucose apart. Then we have the prep reaction. So the prep reaction takes those glucose molecules, prepares it for the next phase, which is the citric acid cycle, and it turns it into, it takes the pyruvate from the glycolysis phase and turns it into coacetyl enzyme A. So, and the prep reaction occurs in the interior of the mitochondria in the matrix. The citric acid cycle also occurs in the matrix, and it uses that coacetyl enzyme A, and it uses those inputs of energy, NADP, FADPH, and it, as reduction and oxidation occurs, it releases hydrogens, it releases electrons. So all of these free-floating hydrogen electrons are building up a gradient within the mitochondria. So our fourth and final phase of cellular respiration is the electron transport chain. So within the electron transport chain, those electrons are excited, they're moved down that chain, and the bulk of our ATP is produced in our electron transport chain. So it occurs in the cristae. So the same way as in photosynthesis, we have ATP synthase. That is the same concept of what's occurring in the electron transport chain. So it's pulling across these membranes the gradient buildup of electrons and hydrogens to produce ATP. So this is important. I guarantee you this next part will be on the test. So I want you to know the phases of cellular respiration, and I just numbered them over here, and those numbers coordinate with the ones we just talked about. And I want you to know how much ATP is produced in each phase. So the first phase is glycolysis, and we produce 2 ATP. Now in glycolysis, if we run low on oxygen, we enter into what is called fermentation, right? Fermentation occurs in the absence of oxygen, but it still produces 2 ATP. So whether we go through glycolysis with an adequate amount of oxygen or we have a reduced amount of oxygen, we still are producing 2 ATP, which we would have a reduced amount of oxygen in a situation like grain in a silo, in a silage bag, and we'll kind of talk about that on the slides for our next unit. We actually talk about forage production. So our second one is the prep reaction. The only purpose of the prep reaction is to take that pyruvate, turn it into coacetyl enzyme A. It uses a little bit of energy, but it produces none. So the output of ATP in the prep reaction is 0. So the citric acid cycle occurs, takes that coacetyl enzyme A, reduction, oxidation, breaks that apart. We have those hydrogens, electrons that are free floating, and it has a total output of ATP of 2. So far that's not a whole lot of ATP, right? So all of the power in cellular respiration, all of our ATP production comes from our electron transport chain. The electron transport chain, it differs from organism to organism, is 32 to 34 ATP produced. Giving us a total ATP of 36 to 38 produced in cellular respiration per 1 molecule of glucose. So remember, 1 molecule of glucose, we're getting 36 to 38 ATP. So what I also want you to remember about cellular respiration and photosynthesis, so we discussed the equations for each and how they're very similar. So I drew them out here for you. So here's our equation for photosynthesis. Remember, for photosynthesis, we have an input of energy in the form of the sun. Cellular respiration is the equation for photosynthesis, but it's opposite. Instead of our output being our glucose and our oxygen, we have an input of glucose and oxygen in cellular respiration. We have an output of water and carbon dioxide, where in photosynthesis we have an input of water and carbon dioxide. And we have the production of energy. So photosynthesis uses an input of energy in the form of the sun. Cellular respiration produces an output of energy in the form of ATP, okay? Oh, and while we're looking at this, and you can go back, remember that reduction or redox reaction occurs over and over again in photosynthesis and in cellular respiration. So if we just look at our photosynthesis, so if we have a loss of a hydrogen and a loss of an electron, that is reduction. And then if we have a gain of a hydrogen and a loss of an electron, or a gain of a electron, sorry, that is oxidation. And we have the same thing occurring in cellular respiration. We have a redox reaction happening. All right, and then we talked about cell reproduction. So we talked about mitosis and meiosis. So what I want you to remember with mitosis and meiosis is cell growth. Happens all the time. Every time a plant puts on a new leaf, every time a plant has a flower, every time it makes a seed, mitosis is occurring. Meiosis occurs only for sexual reproduction. So it only occurs at a specific time when the plant is mature, and it happens only in specific areas or regions of the plant. So meiosis is occurring either in the gamete. Sorry, we had a mishap. So mitosis is cell growth, happens all the time. Meiosis is cell reproduction, only happens at specific times. So remember, the stages of mitosis are the same. The stages are the same in meiosis, and we can remember those by saying P-MAT. So this first stage is prophase. So if we were to look at the cell under a scope, in prophase, what's happening is our nuclear envelope is dissolving and our DNA is replicating. During metaphase is we have these polar ends called an axis, and we have spindle fibers that form and go through the middle of the cell. And the chromosomes line up along those spindle fibers straight down the middle of the cell. What happens in anaphase? In anaphase, our spindle fibers start to retract and it pulls those chromosomes apart. So if you remember back or review back to that slide where I showed you the actual picture of the chromosome, remember the center of the chromosome is called the centromere. When it is still connected by one centromere, and it's called a daughter chromosome, that's what we have in metaphase. In anaphase, when they're pulled apart, we end up with 2 centromeres and 2 individual chromosomes, and they're called sister chromatids. And that is what we actually have during anaphase. So in telophase, we have the nuclear envelope has started to reform in telophase. So it reforms around each one of the clusters of chromosomes. And then we have a process at the very end of telophase called cytokinesis. And cytokinesis is when the plant cell will create a cell wall through the plasma membranes of little vesicles will actually join and create a barrier through the center of the cell. So in cytokinesis, at the end of cytokinesis, we actually have 2 individual cells. With a fully formed nuclear envelope back around your DNA. So, and I just wanted to then touch on a few things that we had talked about. So we talked about genotypes and phenotypes. So remember, our genotype is the expression of that gene. So if we write it out, if we're breeding for something that is tall, or breeding maybe for it to be short, how we write that out is our genotype. So big T would be tall, little t would be short. So when we talk about our phenotype, that is visually what we see as the outcome. So when we see the plant as being short, or we see the plant as being tall, we can then say what the actual genotype of that plant is based on the phenotype, what we visually see of the plant. Let's see, let me see if I can remember anything else. Oh, we talked about diploid and haploid, so don't forget to review that cell again. And is Dad taking my car? Yeah. Get my purse out, please. So if something is haploid, we say that it is 1N, and remember diploid is 2N. So in mitosis, a 2N cell produces another 2N cell. So it is growth of the cell. When we talk about meiosis, the full purpose of meiosis is the reduction in chromosome number. So at the end of meiosis, instead of having 2N cells, we have 4 haploid or N cells. So if you forgot that whole process, go back to that slide and review that slide. We also talked about the definition of breeding, and at the very end of that chapter we talked about some of genetic modification that we can do on plants and that definition. So go back to the very end of that genetics PowerPoint and make sure that you review those individual definitions that are on those slides. So now our last unit is the one on propagation, and it's the one that we didn't get to talk about in class. It's the slides that I posted. You should have hopefully already done your worksheet questions, your quiz, I guess, for that. So I had a lot of questions and emails about the fact that the quiz kept counting question number 2 wrong. And that is something on my end. Blackboard is very touchy about how you enter in an answer for a quiz when it's grading it electronically. So during propagation, we have 2 forms of propagation. We have sexual propagation and asexual propagation. Sexual propagation is the production of seeds. Go back to that slide and look at those definitions of breeder seed, foundation seed, certified seed, and just kind of review what order those go in, and who's the direct recipient of that seed in the end. And then asexual propagation, that is vegetative reproduction. And in your slides, the reason I wanted to go over this is very important. So there are 3 structures that are formed with vegetative propagation. Adventitious roots, adventitious shoots, and a callus. What that means is that in a vegetative cutting, you can literally take a cutting of the end of a plant, a leaf like this. And what will happen is if you put this in soil, you will get root growth that originates from the stem, doesn't originate from previous roots. You will get a stem that can originate from a leaf. So it can originate from tissue that is not like it. And then a callus is when you take a cutting like this, a callus will actually form on the very end of that tissue. And a callus is a cluster of cells that can differentiate into other tissues. So your callus is either going to differentiate into a root, a stem, a leaf, but it is some other tissue other than the tissue that it came from, so other than a like tissue. In that propagation, also there's talked several slides in there about different methods of propagation, and some talking about making a runner, making a graft. Just sort of review those and know that those are different ways to vegetatively propagate, but I wouldn't don't concentrate too much on that. Some of that might be on the test, but that won't be the bulk of the test. So just kind of review those vegetative ways to actually propagate something by a division, and I think that's all. So if you guys listen to this, go over your review sheet that I posted on Blackboard, and watch Blackboard for information about the test on Tuesday. Remember, our test is this coming Tuesday. I'll open it up on Blackboard for you to see it. So, or hopefully that's the plan. This will be my first full test to put on Blackboard, so give me some slack if it doesn't exactly look right, and if I end up kind of like on the quiz getting a question that's continually counted wrong. I'll be able to see that. I think it sends me a report, so I'll know if everybody misses question 2. It's just something to do with Blackboard. So hope you guys are doing well. I really miss seeing you in class, and maybe I'll make another video again so that we can actually kind of talk face to face. So see y'all later. Bye.